L forms. L forms, also known as L phase variants, are cell wall defective organisms. L forms, also known as L phase variants, are cell wall defective organisms, are abnormal in pleomorphic variants of bacteria that lack a rigid cell wall but still retain the ability to grow and multiply. Here are the detailed characteristics and mechanisms and clinical significance of the L forms. Clinical significance of L form. L forms, also known as L phase variants, are cell wall defective organisms, are abnormal pleomorphic variants of bacteria that lack a rigid cell wall but still retain the ability to grow and multiply. Here are the detailed characteristics and mechanisms and clinical significance of L forms. Discovery and naming. L forms were first discovered by E. Kleinerberger, Nobel Prize winner in 1935, while studying Streptobacillus moniliformis. She named these abnormal growth forms L forms in honor of the Lister Institute in London, where the discovery took place. Discovery and naming of L form were first discovered by A. E. Kleinberger, Nobel in 1935, while studying the Streptobacillus moniliformis. She named these abnormal growth forms L forms in honor of the Lister Institute in London, where the discovery took place. Formation and causes. Formation and causes. Because they lack a rigid cell wall, L forms do not possess a regular shape or size, pleomorphism, and become roughly spherical. They can emerge in two ways. Spontaneous, they can arise naturally in bacteria like Streptobacillus moniliformis and bacteroid species. Next, induced. Induced, they can be artificially induced by exposing a normal bacteria to agents that inhibit cell wall synthesis, such as penicillin, or by placing them in a synthetic media with hypertonic osmolarity, that high salt concentrations, to prevent the cells from bursting. So, spontaneous, they can arise naturally in bacteria like Streptobacillus moniliformis and bacteroides species. Induced, they can be artificially induced by exposing normal bacteria to agents that inhibit the cell wall synthesis, such as penicillin, or by placing them in synthetic media with a hypertonic osmolarity, that means in high salt concentration, to prevent the cells from bursting. Types of L forms. Types of L forms. Stability. L forms are more stable than protoplast and spiroplast and are categorized based on their ability to revert back to their original bacterial shape. The unstable L forms, these variants can revert to their normal cell wall containing bacillary form once the inducing stimulus, that is penicillin, is removed, allowing them to resume normal cell wall synthesis. Next, stable L form. Stable L form, these permanently continue the cell wall deficient state and do not revert even when cultured in environments free of cell wall inhibitor. Despite lacking a cell wall, stable L forms remain biochemically and antigenically identical to their parent bacteria. Cultivation characteristics of L forms. Cultivation characteristics of L forms. L forms cultivation characteristics are difficult to cultivate but are capable of growing on a suitable solid nutrient medium, provided the medium has the correct osmotic strength on agar media. They produce small colonies that have a distinct fried egg appearance, an opaque central zone of growth embedded within the agar, surrounded by a translucent peripheral zone on the surface. Clinical significance. Clinical significance and antibiotic resistance of L forms. Clinical significance and antibiotic resistance of L form. While L forms are generally considered non-pathogenic to laboratory animal and may not be directly initiate a disease on their own, they pose special problems in medicine, like antibiotic resistance, because they completely or partially lack a cell wall. L forms are naturally resistant to antibiotics that target cell wall synthesis, such as penicillin. Persistence and relapse. L forms play a critical role in the persistence of chronic infections like such as pyelonephritis. During antibiotic therapy, normal bacteria may transition into L form to survive the drug. Once the antibiotic treatment ceases, unstable L forms can revert to their virulent original bacterial form, leading to a relapse of the disease. Difference from mycoplasma. Difference from mycoplasma. L forms share several visual similarities with mycoplasma. L forms having several visual similarities with mycoplasma. A naturally cell wall less genus of bacteria. Mycoplasma is a naturally cell wall less genus of bacteria, such as the fried egg colony appearance and a lack of peptide glycan. However, they differ in several key ways. Unlike mycoplasma, L-form do not require sterols for growth and plasma membrane stability. L-forms are generally not filterable, whereas mycoplasma are. L-forms still remain measurable remnants of cell wall component, while mycoplasma completely lacks them. Reproduction of L-forms is inhibited by penicillin, whereas mycoplasma reproduction is not. So this is all about L-forms.